Have your boots been under?
For the second part of the experiment, instead of recording from the sensory nerve, we'll be stimulating from the sensory nerve. So we move the recording electrode up to the connective nerve, and we take the stimulating electrode and place it onto the circle nerve. From the first part of the experiment, we cut the circle nerve, and so this time we'll go to the other side on the other circle nerve. And if this is the sixth ganglion, you'll want to cut far away from it so that we have plenty of space to suck up into the electrode for stimulation. This electrical stimulation will be a better and more standard stimulation than the air pulse we were using before, and will be forced up the nerve into the connective nerves that we'll be recording from. For stimulating the prep, you can use a stimulator, or you can use this internal stimulation found in the A to G board. For recording this part of the experiment and the stimulation, we'll be using the scope program. You should again use your lab manual for setting up the particular settings for the procedure. Now we're looking at a stimulation of the circle nerve up to the connective nerve through the neural circuit. We can change the voltage on either the program on the computer or through the stimulator until we find a response. Once you find the response, you can look at pharmacological influences. Now I can look at the synaptic properties by dissecting out the chemical and electrical synapses of the neural circuit. From this experiment, you've learned how to dissect the cockroach, take extracellular recordings, and data analysis. From a follow-up experiment, you'll get to see the anatomy of the neurological circuit through the use of dyes.
Now you've learned how to record electric activity from the cockroach neural circuit, then we can go ahead and switch the solution with different solutions that contain different pharmacological agents like agonists and antagonists, and it will answer our question that we addressed at the beginning.
Reproducing these classical results should be pretty easy. Challenge now is to come up with new applications for these experiments and new questions to ask.
Yeah.
Have fun with your experiments.